Hello students, welcome back to my biopsychology class. In this video, we'll be talking about the chemical senses. So let's take a look what we will have in this video. Firstly, we will overviewing about the taste and olfaction, and then we briefly will be discussing about pheromones and lastly, synesthesia. All right, so as you might know that vision, hearing, and touch provide us information that is useful for many purposes, right? But taste is unique. Taste is actually useful for just one function. It will telling us whether to swallow something or to spit it up, yeah? And this function is actually more important for some species than for others. And also taste results from stimulation of the taste buds, the receptor on the tongue. And when we talk about the taste of food, we generally mean flavor, yeah, which is a combination of taste and smell. Whereas other senses remain separate through the cortex, taste and smell axons actually converge onto many of the same cells in area called the endopiriform cortex. And it also converge enables taste and smells to combine their influences in food selection. All right, the receptors for taste actually are not true neurons, but it is like modified skin cells, yeah? But like neurons, taste receptors have excitable membranes and release neurotransmitters to excite neighboring neurons, which in turn then transmit information to the brain. And like skin cells, taste receptors are gradually sloughed off and replaced, and each one lasting only about 10 to 14 days. And mammalian taste receptors are in taste buds, yeah, located in the papillae on the surface of the tongue, as you can see in this figure. A given papillae may contain up to 10 or more taste buds, and each taste bud contains about 50 receptor cells. In adult human, taste buds lie mainly along the edge of the tongue. So you can demonstrate this principle as follows. Try to soak a small cotton swab in sugar water or in salt water or in vinegar. Then touch it lightly on the center of your tongue, not too far toward to the back, yeah. And if you get the position right, then you will experience little or no taste. All right, so how many kinds of taste receptors? Traditionally, people in Western society, they have described tastes in terms of sweet, sour, salty, and bitter. However, some tastes defy categorization in terms of these four labels, right? And then the question is, how could we determine how many kinds of taste do we have? One way to identify taste receptor types is to find procedures that alter one receptor but not others. For example, here, you may try to chew a miracle berry. It's kind of berry, it's native to West Africa. It gives little taste itself, but temporarily will change sweet receptors in our receptors. So actually, miracle berries here contain a protein called miraculin that modifies sweet receptors and enabling acid to stimulate them. So if you try miracle berry extract, which you can find in the internet, so anything acidic will taste sweet in addition to its usual sour taste for the next half hour. Some people use this extract as diet acid so they can get sweet taste without the calories. And further evidence for separate types of taste receptors comes from the studies of the following type. Now you can also try this by yourself. You can soak your tongue for 50 seconds in a sour solution, such as unsweetened lemon juice. Yeah, then try tasting some other sour solutions, such as dilute vinegar. And after you did this, you will find that the second solution tasted less sour than usual and perhaps not sour at all. And this phenomenon called adaptation. It reflects the fatigue of receptors sensitive to sour taste. And then now try tasting something salty, sweet, or bitter. These substances taste about the same as usual. In short, you fail to show cross-adaptation or the reduced response to one taste after exposure to another. Evidently, the sour receptors are different from the other taste receptor. Similarly, you can show that salt receptors are different from the other and so forth. And although we have long known that people have at least 4 kinds of receptors that we've talked that before, several types of evidence then suggest a fifth. Is the glutamate, as in monosodium glutamate or MSG. Yeah? So the tongue has a glutamate receptor that resembles the receptor of glutamate as a neurotransmitter. And glutamate tasted somewhat like unsalted chicken broth, right? And the English language had no word for this taste, so English-speaking researcher then adopted the Japanese word umami, or perhaps in Bahasa Indonesia would be like gurih, yeah? Also, perhaps we have a 6th type of taste, yeah? Is the taste of fat, what it is. So when people taste long-chain fatty acid, they say the taste is not sweet, not sour, salty, nor bitter, or only slightly resembles umami. And researchers did their best to make sure people were responding to a taste experience and not just a texture. And then they suggest that the term oleogestus for the taste of fat. All right, so the mechanism of taste receptor. So the first one, the saltiness receptor is actually, it's the most simple, yeah? It recalls that a neuron produces an action potential when sodium ion cross its membranes. A saltiness receptor, which detects the presence of sodium, simply permits sodium ions on the tongue to cross its membrane. And chemicals that prevent sodium from crossing the membrane weaken the salty taste. And sour receptor detect the presence of acid, while sweetness, bitterness, and umami receptors resemble the metabotropic synapses that we have discussed in chapter 2. So after a molecule binds to one of these receptors, it activates a G protein that releases a second messengers within the cell. People have 2 types of sweetness receptor and 2 types of umami receptors, each with somewhat different sensitive. And bitter taste used to be puzzled because bitter substances include a long list of dissimilar chemicals. The only common factor is that they are to some degree toxic. So what receptor could identify such a diverse set of chemicals? The answer is that we have not one bitter receptor, but a family of 30 or more bitter receptor, and each respond to a few related compounds. One consequence of having so many bitter receptors is that we detect a great variety of dangerous chemicals. The other is that because each type of bitter receptor is present in small numbers, we don't detect very low concentration of bitter substances. And many bitter chemicals also trigger receptors in the nose, yeah? It will provoking coughing and sneezing if you happen to inhale them. That is, bitter chemicals are toxic and the body will do anything it can to expel them. And then how then taste code in the brain? So information from the receptor in the anterior 2/3 from the tongue travels to the brain along the chorda tympani. It's a branch of the 7th cranial nerve or in the facial nerve, yeah. And taste information from the posterior tongue and the throats travel along the branches of the ninth and 10th cranial nerve. And what do you suppose would happen if someone anesthetized your chorda tympani? You would no longer taste anything in the anterior part of your tongue, but you probably will not notice because you will still taste in the posterior part. However, the probability is about 40% that you would experience taste phantoms. It's analogous to the phantom limbs experience discussed in chapter 4, yeah? That is, you might experience taste even when nothing was on your tongue. Evidently, the inputs from the anterior and posterior part of your tongue interact in complex ways. And the taste nerve projects to the nucleus of the tractus solitarius, or the NTS. It's a structure in the medulla. And from the NTS, information branches out, reaching the pons, the lateral hypothalamus, the amygdala, the ventral posterior thalamus, and 2 areas of the cerebral cortex. One of these areas, the somatosensory cortex, will respond to the touch aspect of tongue stimulation, and the other area, known as the insula, is the primary taste cortex. The insula in each hemisphere of the cortex receives input from both sides of tongue. A few of the major connections are illustrated in this figure, and certain areas of the insula are dominated by cells responding mainly to sweet tastes, whereas other areas are dominated by cells responding to bitter. And it is easy to assume that foods taste the same to someone else as they do to you, right? And if we ask people to describe how strong various foods taste, they agree on which foods have bland, strong, or very strong flavors. However, when you rate some food as tasting very strong, you mean that it's strong compared to your other taste experience, which might be stronger or weaker than someone else's. In fact, some people have 3 times as many taste buds as other people do on their fungiform papillae near the tip of their tongue. That anatomical difference depends partly on genetics, but also on age, hormones, and other influences. Women's taste sensitivity varies with their hormone and reaches its maximum during their early pregnancy, when estradiol levels are high. So that tendency is probably adaptive. That during pregnancy, a woman needs to be more careful than usual to avoid harmful foods. And people with more taste buds, known as super taster, tend to dislike strongly flavored food, especially food that tastes very bitter to them, but only mildly bitter to other people. People at the opposite end, having the fewest taste buds, tolerate many somewhat bitter foods. A demonstration sometimes used in biology laboratory classes is to taste the phenylthiocarbamide or PTC or the 6N propyluracil or PROP. And most people taste low concentration as bitter, but people with the fewest taste buds, misleadingly known as non-taster, taste it only at high concentration. One gene controls most of the variants, although other genes contribute as well. So if you would like to classify whether you're yourself a taster, non-taster, or super taster, you can follow the instruction in this table. All right, let's move on to the olfaction. So olfaction is the sense of smells, right? And it is a response to chemicals that contact the membranes inside the nose. For most mammals, olfaction is critical for finding foods and mates, and also for avoiding dangers. For example, rats and mice show an immediate, unlearned avoidance of the smells of cats, foxes, and other predators. Those smells also cause them to release stress hormones. Mice that lack the relevant olfactory receptor fail to avoid the predators. And people with certain disease have a characteristic unpleasant odor, and it makes people who avoid that odor decrease the risk of contagion. And olfaction is also especially important for our food selection. Much of the flavor of food is really based on its odor. So try holding your nose while you eat and notice how much flavor then you will lose. And olfaction is also plays an important role in social behavior, as you may heard the expression of smell of fear. And actually, research support that idea. So an experimenter collected sweat from young men's armpit, while the man watched video that evoked fear, disgust, or no emotion. And later, the researchers recorded facial expression by young women who sniffed the samples. And women smelling the fear samples showed a mild fear expression, and those who smelled the disgust sample looked disgusted, and those smelling the neutral sample showed little or no expression. Evidently, smells give us a clue to how someone else is feeling. So if you were exposed to the smell of other people with no other information about them, and you rated their desirability as a potential romantic partner, you would probably prefer people who smell a little different from yourself and your family members. Why? Because avoiding a mate who smells like your brother or sister reduces the chance of inbreeding. It also increases the probability that your children will have a good variety of immunities because chemicals from the immune system contribute to body odors. Curiously, when women start taking contraceptive pills, their preference for a different smelling mate decreases. One speculation is that the risk of inbreeding is unimportant for women who cannot become pregnant at that moment. And it's also olfaction is also related to current infectious disease, COVID-19. Anosmia is one of neurological symptoms of COVID-19 infection. And anosmia here is the loss of olfaction. It means that an individual cannot smell or taste it. And more than half of COVID-19 patients reported having anosmia, while this anosmia is a temporary symptom in COVID-19 patient. And we will moving on to the olfactory receptors. Researchers here estimate that people can distinguish among more than 1 trillion olfactory stimuli. And the neurons responsible for smell are the olfactory cells that line the olfactory epithelium in the rear of the nasal air passage that you can see in the figure. And in mammals, each olfactory cell has cilia or thread-like dendrites that extend from the cell body into the mucous surface of the nasal passage. And olfactory receptors are located in the cilia. And how many kinds of olfactory receptors do we have? Researchers identified a family of proteins in our olfactory receptor, as you can see in this figure. It's like the metabotropic neurotransmitter receptors. Each of these proteins traverses the cell membrane 7 times and responds to a chemical outside the cell by triggering changes in a G protein inside the cell. And the G protein that provoke chemical activities that lead to an action potential. And the base estimate is that humans have several hundred olfactory receptor proteins, whereas rats and mice have about 1,000 types. And when an olfactory receptor is stimulated, its axons carries an impulse to the olfactory bulb, yeah? And also the receptors sensitive to a particular chemical are scattered haphazardly in the nose. Their axons find their way to the same target cells in the olfactory bulbs, such the chemicals of similar smell excite neighboring areas and chemicals of different smell excite more separated areas. And the olfactory bulb send axon to the olfactory area of the cerebral cortex. And a complex substance such as food, it activates a scattered population of cells. And many cells then respond strongly to a particular kind of food, such as berries or melon. Yeah? And as in the olfactory bulbs, chemicals that smell similar to us evoke activity in neighboring cells. And olfactory receptors, actually, they are vulnerable to damage because they are exposed to the air. And unlike your receptors for vision or hearing, which remain with you for a lifetime, an olfactory receptor has an average survival time of just over a month, yeah? And at that point, a stem cell matures into a new olfactory cell in the same location as the first and expresses the same receptor protein. And people vary in their sense of smell more than you might guess. Most of the genes controlling olfactory receptor have variant forms, and on average, 2 people chosen at random probably differ in about 30% of their olfactory receptor genes. And compared to anyone else you know, you probably experience some smells as stronger, some as weaker, some as more pleasant, and some as less pleasant. In addition to individual differences based on genes, odor sensitivity declines with age. The declines then also varies among others. Women detect odors more readily than men at all ages and in all cultures that researchers have tasted. In addition, young adult women gradually become more and more sensitive to a faint odor they repeatedly attend to, and until they can detect it in concentration on 1/10,000 of what they could at the start. Also, men, girls before puberty, and women after menopause do not show that effect. So it apparently depends on female hormones that we can only speculate on why we all hold a connection between female hormones and odor sensitization. And additional senses is important for most mammals, yeah. Although it's less so for humans, is the pheromones here. So the vomeronasal organ, or VNO, is a set of receptor located near but separate from the olfactory receptor. And unlike olfactory receptor, the VNO receptors respond only to pheromones. It's the chemicals released by an animal that affect the behavior of other member of the same species. So for example, if you have ever had a female dog that was neutered whenever she was in her fertile period, even though you kept her indoors, your yard attracted every male dog in the neighborhood that was free to roam. And each VNO receptor responds to just one pheromone in concentration as low as 1 part in 100 billion. Furthermore, the receptor does not adapt to a repeated stimulus. So let's say, have you ever been in a room that seems smelly at first, but not a few minutes later? So it means that your olfactory receptor respond to a new odors, but not to a continuing one. VNO receptors, however, continue responding even after prolonged stimulation. And in adult humans, the VNO is tiny and has no receptor. It is vestigial, that is a leftover from our evolutionary past, actually. Nevertheless, part of the human olfactory mucosa contains receptors that resemble other species' pheromone receptors. And the behavioral effect of pheromone apparently occur unconsciously. That is, people react to certain chemicals in human skin even when they describe them as odorless. So let's say here the smell of a sweaty woman increases a man's testosterone secretion, especially if the woman was near her time of ovulation. And this effect is stronger for heterosexual men than for homosexual men. And the smell of a sweaty man actually does not increase sexual arousal in women. Instead, it increases release of cortisol, the stress hormone. Evidently, a man reacts to a sweaty woman as a sex signal, and a woman reacts to a sweaty man as a potential danger signal. And the best documented effect of a human pheromone relates to the timing of women's menstrual cycle. Yeah, according to several, but not all, reports, women who spend much time together find their menstrual cycles become synchronized, unless they are taking birth control pills, And another study dealt with the phenomenon that a woman in an intimate relationship with a man, they tend to have more regular menstrual periods than women not in an intimate relationship. And according to one hypothesis, the man's pheromones promote this regularity. In the study, young women who were not sexually active were exposed daily to a man's underarm secretion. I know it's disgusting, and getting women to volunteer for this study wasn't easy, right? And gradually, over 14 weeks, most of these women's menstrual periods became more regular than before. In short, human body secretions probably do act as pheromones, although the effects are more subtle than in most other mammals. All right, finally, let's consider something that is not one sense, but a combination of senses, It's the synesthesia. So synesthesia is the experience some people have in which stimulation of one sense evokes a perception of that sense and another one also. So for example, someone might perceive the letter G as green, or say that each taste feels like a particular shape of the tongue, In the words of one person, so for example, a person with synesthesia, she or he tell the researchers that, to me, the taste of beef is dark blue, or to me, the smell of almond is pale orange. And when tenor saxophone play, the music looks like a floating, suspended, coiling snake ball of lit up purple neon tubes. All right, that's pretty odd, but it happened. And one person with synesthesia was able to find the number two, consistently faster than other people, explaining that he just looked for a patch of orange, yeah, in this experiment. So what is cautious synesthesia actually? So it clusters in families suggesting a genetic predisposition, yeah, and it frequently occurs in the same family as people with absolute pitch, yeah, suggesting that the two conditions share a genetic predisposition. However, obviously people are not born with a letter to color or number to color synesthesia, and no one is born knowing the letter of the alphabet, yeah. So, but in some cases, we see where people learn their association, yeah. Researchers found 10 people with synesthesia whose association nearly matched the color of Fisher-Price refrigerator magnets. It's very interesting. And various studies attest to the reality of synesthesia. People reporting synesthesia have increased amount of gray matter, yeah, in certain brain's area and altered connection to the other areas. And people who perceive colors in letters and number have increased connection between the brain areas corresponding to colors and those responding to letters and number. And when people misperceive a stimulus as an illusion, the synesthetic experience corresponds to what the person thought the stimulus was, not what it actually was. And this result implies that the phenomenon occurs in the cerebral cortex, yeah? So it's not in the receptor or their first connection to the nervous system. And one hypothesis is that the axon from one cortical area branches into another cortical area. And this explanation does apply to at least some cases. So for example, one woman suffered damage to the somatosensory area of her right thalamus. Yeah, initially she was, as expected, insensitive to touch in her left arm and hand. And over a year and a half, she gradually recovered part of her touch sensation. However, during that period, the somatosensory area of her right cortex was receiving little input, and some axon from her auditory system invaded the somatosensory cortex. And as a result, she developed an unusual auditory to touch synesthesia. Many sounds causes her to feel an intense tingling and sensation in her left arm and hands. All right, that's all about the chemical senses. Thank you for your attention. I'll see you again. Take care, everybody.